Happy World Ocean Day. We're celebrating today. I hope you saw our Instagram reel already floating around on social media about the beauty of the Galapagos Islands and how we are working towards conservation. With this, we also wanted to share with our public about the recent discoveries that we've had here in the Galapagos Marine Reserve. So we invite you to dive into the Galapagos Marine Reserve with our deep sea scientists, Stuart Banks and Salome Buglas. She will be joining us in a few more minutes. So first of all, I wanted to share the work of the Charles Darwin Foundation. Our new mission is to tackle the greatest threats and challenges to Galapagos through scientific research and conservation action in order to safeguard one of the world's most important natural treasures. So today we will be co-hosting, me as Camila Bermudez. I am the communications officer, and Lisa Nagore. You want to say hi? Yep.
Yes. So I have been working with CDF for 5 years in the fundraising department. So it's a pleasure to be here and co-host this webinar with our talented scientists.
Okay, so our expositors will be Stuart Banks, our oceanographer. He is one of the 2 scientists who recently discovered the Galapagos' first recorded deep sea coral reef. It was on board the RV Atlantis and its human submersible Alvin. This happened between March 28 up until April 22nd. He was also on board with Salome Buglas. She was also a participant into this month-long expedition that brought us this great news that he will be sharing shortly. After Stuart, Salome will be presenting as well. She's our marine scientist. She discovered and studies the tropical kelp forest sites across seamount summits and coastal drop-offs in the Galapagos Marine Reserve as well. So she will be sharing with us her recent discoveries, and will also be helping answer all the questions about the month-long discovery that she shared with Stuart.
Perfect. Thank you, Camila. So all the work of the Charles Darwin Foundation, including the research that is going to be presented today by Stuart and Salome, depends entirely on the support from our generous donors. So therefore, we created this special campaign for the webinar to fundraise for one of the data analysis workstations that the project currently needs to analyze a large amount of data that they collected during the most recent expeditions. Our goal for this webinar is to raise $2,500, and you can donate by clicking on the link that I just put in the chat box. But we will also share it with you in the email that we will send after our webinar. So the campaign will stay open until next Friday, which is June 16th. So Camila, if you go to the next slide, every donation counts. So even if we don't meet our goal, all the donations will support the project, going either towards fieldwork, lab supplies, or hopefully for the data analysis workstation. And before jumping into the presentations, just some housekeeping. We will have time for questions at the end of both presentations. So you can put your questions into the Q&A box below, and we will try to answer as many as we can, or all of them if the time permits. And also this presentation is being recorded, so we will share the recording with you after the webinar. So I think with that, we can jump right in, right, Camila?
Yes, sure. Stuart, you want to share your screen? So
yeah, okay, thanks. Let's see. Camila, if you could just have a quick look, it says host disabled participant screen sharing.
There you go.
I think you may need to enable it.
Yeah, I think you can do it now.
Okay, yeah, perfect. Okay, can you see that? Are we good? Can you hear me?
Yes, we're good.
Okay, great. Okay, well, good morning, everybody, and thank you very much for taking the time to spend with us this morning. World Ocean Day. I mean, every day should be World Ocean Day, right? You know, the oceans are so amazingly important as part of the life support system that we have as people on the planet. Here in Galapagos, you know, we've been working in Science for Conservation through the station with our partners for the last over 30 years, with many, some of which I also saw who are on this call with us in this webinar this morning, who have worked tirelessly towards trying to ensure that we can provide the best technical advice for the conservation of some of the last kind of amazing pristine habitats that we have, working how we as people can better interact with the resources and this amazing kind of biodiversity that we have in the oceans. About today is a new area for us, but it's actually something that we've had on the radar as important research for conservation for the last 20 years. And it's only recently that with the advent of new technology, we've started to have access to the deep systems, you know, below and beyond the area that we can easily reach through recreational and technical diving in the near coast. So that's kind of what we'll be talking about today. And it's a nice mix between what I'm going to present is part of the work which was possible thanks to strategic collaboration with various universities, Woods Hole in the US, Woods Hole Oceanographic Institution, the NSF operation of the Atlantis, and the Alvin manned submersible crew, but also universities from the UK, Essex and Bristol. And it's one example of several that we've had in the last 20 years, which has started to give us the opportunity to be able to go deeper beyond, you know, the restrictions that we've had in trying to access these amazing deep water systems. So a good place to start is there's a lovely video which was put together. Some people here you may recognize, and they say kind of a picture is worth 1,000 words. So I think perhaps we'll start with this.
Y estamos ahora en la última parte de un crucero de casi 22 días explorando las profundidades de Galápagos usando una tecnología nueva sumersible.
The coral reef that we've discovered on this expedition is a deep sea coral reef. So we found it where below the depths where light disappears and animals are existing from all of the material that falls down, it rains down. It's called marine snow. It was unusual for many reasons, but it was just so full of life.
On most volcanic islands, the farther out and the deeper you go, the older the rocks are. And one of the problems we've had, or one of the challenges we've had in looking at the terrestrial parts of the volcanoes is it's like a frosting on a cake. You only see the upper part, you don't know what's on the inside. But as we go deeper and farther out, we can get into the older parts of the volcano. And so we're really excited to see how these volcanoes evolve.
No hemos encontrado arrecifes de este tipo en estas profundidades tan extensos y desarrollados. Tienen por lo menos varios miles de años, si no mucho más, de crecimiento y están en estado pristino, que es muy importante a nivel mundial.
Okay, so So yeah, so maybe to perhaps kind of give a bit of context. Since the Marine Reserve was established in 1998, such as we know it today, as an extension, as you can see in this image here, of 40 nautical miles, covers an area of about 138,000 square kilometers. And recently, through a long process led by Ministry of Environment with various different partners, it's been possible to extend the reserve, recognizing there's this important connectivity between the other marine protected areas in the Eastern Tropical Pacific, which is the extension that you see of the Hermandad Reserve that effectively adds another 60,000 square kilometers. So there's 200,000 square kilometers of open water area surrounding the islands. Historically, we've really only been able to work in the near coast. So in this image that you're looking at the moment, this is the bathymetry that we have across the archipelago. What you're looking at the moment, the red area, which is close to the shore, this is the area which is kind of within our kind of safe diving reach. And it's the area that was kind of a priority for marine conservation and research in the last 20 years, because that's the area where you had the most intervention, you had the most kind of interaction in the earlier years of the reserve between developing fisheries in the coast and tourism. But what we start to see, if you kind of start looking as we move into deeper water across the Galapagos platform, you start to see that if you started to remove the water, you would have this amazing, if not kind of majestic bathymetry and topography, which underlies the islands. This amazing kind of hidden ridges, sunken islands, volcanoes, and a range of different geological features, which basically lend an amazing amount of diversity across the entire archipelago. So as you kind of see these areas which are in yellow here, this is when you're entering what kind of people kind of term it's like the mesopelagic area, or the twilight zone. This is where light can still reach the top of the platform, these yellow areas. And as you start to go down, then you begin to reach the areas where you have very little information. And most of this information is historically collected in previous years from incidental surveys through dredge work throughout the islands, going all the way back to the 1800s with the early Challenger expeditions and the Albatross. And then if we go even deeper, down past 2,000, 3,000 meters, we start to see these amazing terraces along the southwestern edge of the archipelago that then very rapidly in the west drop off to the abyssal plain. There's very few places in the world that have this amazing kind of relief of topography across an oceanographically very dynamic area. And as you know, we're in one of the areas where we have the greatest effect of some of the most kind of varied oceanographic variation within the region, the El Nino phenomenon. So you have this kind of three-dimensional underwater environment covering huge areas, which has never been properly documented or folded into conservation planning for the archipelago. There's only in 2014, 2016 that it was possible through the new management plan with the National Park Service in Galapagos to begin to incorporate and integrate open water areas with the coastal areas within the reserve. And based on that, the station and partners have begun to try to characterize and understand what we have in these different ecosystems to properly represent the biodiversity that they bring to the unique our unique understanding of the archipelago as a near pristine and protected environment. So to do that, obviously, you start to rely on technology, which there's some technology which is cheaper, which you can actually use small ROVs, which you can use to reach kind of these down to about 300 meters. And you start to have problems with managing these are run by small kind of robots called remote operated vehicles. And there's a whole range of these different operations. And the smallest level you can use them to get down to kind of maybe 100 meters. The kind of cost of these operations go up. So in that sense, it's been hugely important to collaborate with ocean expeditions and foundations, such as the Nautilus in previous years, with the the Alusha operation, and various others who have kind of started to contribute to these glimpses of the deep water environment. And Salome will talk more about this afterwards, but one of the other opportunities with using this kind of technology is it has let us look at some of the inshore areas which are just beyond operate. It's one of those rare opportunities where you can say that it was a truly kind of interdisciplinary, transdisciplinary crew. The operation itself is led by and organized by Woods Hole Oceanographic Institution. And it was through the collaboration between Essex University, Bristol University, National Park Service, and INOCAR, the oceanographic branch of the equity, that we were able to bring a range of different kind of focus and disciplines to the same cruise. And all of this was built around the kind of sampling that was possible through the manned submersible, and also by using a series of specialized instruments which are built into the operation of the Atlantis. The Atlantis itself has about 20 to 25 operational crew members, and it can accommodate a science crew of about 25 people. So these cruises kind of tend to be very well designed. In this case, this was an example of a cruise that was originally planned almost 4 or 5 years before. But because of COVID, it was among various other cruises which were planned in the last couple of years, not possible. So this is the first time this year that we've been able to start to return to these kind of operations. And this gives us an amazing opportunity to start to collect the baseline data that we need to really kind of get a handle on what we're looking at. This is actually bathymetric data, which is collected through, it's called a multi-beam, which is a way of collecting high-profile bathymetry at depth across the Galapagos platform. In this case, we're looking at multi-beam, which is collected from a previous cruise, from the Melwood expedition from 2010. But this is essentially very similar to what we've been collecting to try to fill the gaps across the marine reserve to understand what we have in the deep habitat. And as you can see, it starts to reveal in much more detail these amazing features of this is actually in the northwest portion of the archipelago, and you start to see these amazing features. This is northwest Darwin Ridge up here. Wolf and Darwin up here is oceanic pinnacles, and you start to see all these other features, truly oceanic seamounts, which are rising from the ocean floor. And these are the areas that we've been prioritizing, given that seamounts, because of their oceanographic characteristics, tend to be particularly productive areas, forcing productive nutrient-rich water up over these underwater mountains. So these features are also hugely important to associations with the charismatic marine fauna and megafauna and different types of marine communities as you go down through different depths as well. So we start to profile different depths while at the same time collecting information about the oceanographic conditions to be able to understand themes like the deep contribution to biochemical cycling and also the formation of these habitats, many of which are probably particularly sensitive deep water marine systems. And as you can see below, you have these complementary data sets which you put together. In this case, you're looking at side scan sonar data, where you have reflectance, which lets you get an idea of the types of seafloor which we're looking at between sand and rocky areas. So as you saw on the video, the wonderful thing about the Atlantis cruise is they brought together geologists with paleoclimatologists, with ecologists, with biologists and deep water coral experts, along with people working in operational oceanography and oceanographers. So it was really kind of a very unique experience to be able to bring together these different perspectives and these different kind of disciplines together within one particular research effort. And as you can imagine, they all kind of complement each other and start to create a rich story about the habitats that we have hidden within the marine reserve. What you're looking at here is another image from a previous cruise, Pillow Lava, which was brought up wrapped in a dredge chain through some of the NSF boat surveys undertaken in the last 10 years. And as you can imagine, there's a lot of technology involved with these cruises, and the Atlantis certainly has a new bathymetric mapping system, which was particularly effective. So kind of the order of the day of people who were working it was that people would be able to work in different shifts, helping collect data, and for 24 hours around the clock within 12-hour different shifts. So that meant that people were able to work between different areas, and it was an opportunity as well to be able to, for people to be able to learn what other kind of disciplines within marine science are working on, and how they might benefit their particular area of expertise. In this case, we're looking at the oceanographic winch station, which controlled most of the operational aspects of lowering some of the oceanographic instrumentation down through up to 3.5 kilometers in depth at sampling stations. So as you can see here, this is kind of an example of the on-deck work. On the left here, you have a CTD rosette. So this is collects information on salinity temperature, and these are Niskin bottles, which are mounted here, and they collect water samples. And those water samples are being collected at different points within the water column to be able to give us an idea of how the properties of the water change with depth. And that's particularly important with us because a lot of the most biodiverse areas around some of these hidden systems are linked to the amount of oxygen which is in the water, and processes which affect the formation of different habitats, particularly those which have a calcareous skeleton, such as deep water corals. So that provides us with a suite of kind of biochemical information, which is then paired with information which is recovered from very specific missions with the Alvin submersible. So what you're looking at here in the center, this is the A-frame which launches the Alvin in the morning. This is kind of the order of the day, that the Alvin would be working for up to 8 hours underwater with one pilot and 2 scientific observers on board every day of the cruise for the 22 days. And we were able to undertake 22 dives successfully throughout the entire cruise across different areas of interest in the reserve. And this is hugely important. Most of the work is when the Alvin returns, and it has a selection of samples which need to be very quickly ordered and analyzed. And we'll talk a little bit about that next. So this is one of the focus areas, which is being led by University of Bristol, which was looking at the collection of fossil coral as a window into the paleoclimate. So the reconstruction of past ocean environments by using methods which use radioisotopes to be able to get proxies of how temperature and the biochemistry of the deep oceans has changed over scales of up to 50,000 years. And different people working within the research group focus on different time periods. What you're looking at in the center here, these are sediment cores, which are taken by pushing through the surface sediment, and layers are taken of these cores, which are then used in different types of analysis to look at properties with nutrient cycling in the deep ocean floor. And you can see here kind of people would be working on these throughout the day as the samples come in, and organizing a lot of these samples, most of which are split between work which is possible within the research station here in Galapagos and in coordination with National Park as our partners, and the Biosecurity Agency exported for collaborations with external partners in different universities around the world. And these are examples of the biological collections, the golden axis gorgonian up here. And then we start to see, this is Desmophyla, which was one of the targets for some of the fossil coral collections and the coral collections, which are also looking at how deep water communities are collected across the marine reserve. Here, this is a sea cucumber collected at about 3,100 meters, which we've never seen before as well. Most of the samples which we collected are accompanied by video imagery. So what we do is we start to put together high-resolution video imagery with the collections that we have to build our taxonomic record, but also start to understand associations and ecological interactions between species. And this here is another image taken in the marine lab. This is a bamboo coral, which is showing one of the properties of many species adapted to deep water environments where light doesn't penetrate from the surface, bioluminescence when it's agitated through bacteria which are within the coral. And in this case, it seems that some people believe that this is a kind of like a burglar response, so that if the coral is attacked by a particular creature, then by producing this kind of light response, it attracts other predators which scare off what would otherwise be attacking the coral. So there's kind of various different kind of insights into functional traits of deep water systems which you've never seen. And even in the surface, this was a pyrosome. These are colonial tunicates, which we found floating in huge numbers, huge blooms of colonial tunicates in the surface around an area of Isabella, which is called the Oreja de Burro, or the donkey's ear, in the northwest corner of Isabella. And as you can see, these kind of passively floating, filtering tunicates also bioluminesce in the night. So this is just kind of an example of the opportunity of these cruises to be able to collect information that, even within 25 years, is one of the first time that I've actually seen these huge blooms within the archipelago. So the operation of Alvin itself, Alvin is now rated to go down to 6,500 meters. And when we'd actually go out, this is the front of the submersible. And this is hugely important. This is where the different instruments, you can see there's a capacity to be able to vacuum up samples here. And then you have two arms. And importantly, there are different sampling containers. And these sampling containers are where we register where particular samples or rocks with biological specimens are placed during the course of the dive. And you have the two arms here, which are manipulator arms, which are pretty versatile. And then a series of high-resolution cameras mounted around the front of the submersible. What this means is that every night while we were doing the bathymetric mapping, it was possible to map out areas of interest and be able to look at particularly interesting features. One of the problems is there are so many different features which we don't know. But we start to get representative kind of glimpses, if you like, over these 8-hour periods of what's in different kind of habitat configurations around the archipelago. And what you're actually looking at here, these are high resolution pathways which were mapped. This is important. I'm just going to mention this kind of area around here between Santiago and Santa Cruz. At 3 o'clock in the morning before one of the dives, this is where we found a feature. It wasn't on any of the bathymetric maps. And this is what was later called the Coral Croissant, or the Cacho de Coral in Spanish, which was a volcanic sunken crater, half lunar feature, which was rising from about 700 meters from the seafloor up to about 400 to 300 meters across its crest. And it was possible, once we'd identified that, to start to design, using that bathymetry, a detailed dive plan for the submersible the following morning, leaving at 7-8 o'clock in the morning, to be able to look at what was along that feature. So here what you're actually looking at is the edge, a track along the edge where we'd plan to take different samples with different kind of objectives throughout those dives. And here you can see this is the inside of the submarine. When you're down, it's pretty cozy when you're down there, but a wonderful experience. And this is what we found. And this was pretty significant for us. And as you saw in the previous video, I mean, so far in Galapagos, we've documented, since the strong El Nino events of 1997 and 1981-82, we documented a worrying decline in surface coral reefs inshore after the bleaching effects of El Nino, so that now the last remaining extensive coral reefs, which actually form a coral framework, are in Wolf and Darwin and in few isolated other spots around the archipelago. And they are conservation priorities for priority protection within the spatial zoning system that we have. What we found here was that once we rose up from about 600 meters to the crest of these ridges along that volcanic crater, we didn't see rock for literally the 3 or 4 hours across the top of the the feature in this dive. And what you're looking at is accumulated coral framework. This is particularly significant because the amount of live coral across the framework was particularly high. So a healthy underwater cold water coral reef, which feeds from organic matter, not from sunlight, but from organic matter which falls through the water column from above. You would consider like maybe 10–15% live coverage to represent a healthy coral framework or healthy coral system. And they're usually kind of patches. And this was a very rare example, which we haven't seen in many other places, where you have we found in places up to 50–60% live coral cover. And that's significant because cold water corals are amongst some of the oldest living organisms on the planet. In some areas, they've been dated, live coral has been dated to be over 2,500–3,000 years old, which is true of a lot of the underwater creatures, a lot of the underwater species and groups that have been documented. And the other amazing thing is that they have this whole kind of ecology and diversity adapted living in these frameworks around them and associated with these different coral stands. This is mostly a coral called Madrepora and also Dendrophilia, which are kind of the yellow spots that you're seeing there, but also mixed with glass sponges. An absolutely amazing experience. And here you can see some of the coral, which the fossil coral, which kind of builds up underneath, which was part of the collections which are being used for the paleoclimate reconstructions. So the cruise is a opportunity to be able to start to target some of these deeper areas, which are kind of beyond the area that we can easily access based in the islands. And we're working on more partnerships. We have other cruises with the Schmidt Ocean Institute, research organizations as well, which will let us build upon a lot of the discoveries, and we'll continue working with Woods Hole and with the external universities and other partners in the region, such as Smithsonian Tropical Research Institution, University of Costa Rica, to begin to piece together the baseline for deep water areas, which represent about 95% of the marine area, of which less than 5% has been properly documented at a global level. So part of the work which we're doing is a collaborative deep water baseline to be able to work to generate reference points, particularly important when we start to think about the impacts of potential extraction or deep water mining, for example, in other areas such as which is kind of a growing industry, given expectations for the use of rare earth minerals extracted from deep oceans. The other work which our research line which we're looking at is also looking at how we can obviously share our data and how these reference points in these near pristine systems which we're discovering are being affected by climate change, and how other sociological processes can affect their health and their resilience. So we start to generate criteria regarding conservation value and looking at the importance of explicitly including references towards protection of deep water systems. But you need to understand what's there to be able to ensure they're properly represented, which is behind most of the international commitments towards protecting at least 30% of all marine areas and representative habitats by the year 2030. The other thing which is really interesting is that it's an opportunity to start field testing new technology. And there are various groups, such as Ocean Exploration League and National Geographic, amongst others, that we're working with and which we're collaborating with and which we're trying to build further relationships with, to start to use things like deep water autonomous drop sensors, which are cheaper, which mean that we will be able to start kind of deploying across different areas. One of the results of having these huge terabytes of video data is that we're also looking to start to apply things like machine learning and artificial intelligence to be able to fast track the identification of interesting features to then be supervised by experts across these areas. And that's a way of trying to kind of accelerate research across the region, standardize and share it. Obviously, with the final intention of building this into management recommendations and policy not just in Galapagos, but also across the Eastern Tropical Marine Area. So that obviously depends on a lot of collaborations. There's a whole history of extremely important counterpart work, which is ongoing with taxonomists across the world, also looking to develop new DNA and molecular taxonomic identification approaches for many deep water species. The morphology is very difficult to distinguish species. And there's new methods, which mean that even degraded samples can produce kind of phylogenetic information that lets you look at connectivity between different areas in the Eastern Tropical Pacific, and adding to our inventory of marine species within the region. And so we return to the need to be able to process all of this data, which is one of the mentions of the workstations, is that, you know, they're fairly kind of demanding computational exercises. So just to finish, you know, Galapagos is not in a vacuum. It's a connected marine area which is interdependent upon the model of marine protected areas which are being set up in the Eastern Tropical Pacific between Galapagos and other areas. And as you can see here in this overlay, the Galapagos Marine Reserve connects now through the Hermandad Marine Reserve to the Cocos marine protected areas, and the mosaic areas being proposed between Colombia, Costa Rica, and Panama. So this is where we'll be working and trying to build up further collaborations to be able to complement other migratory species work and oceanographic and climate sampling across the entire region, so that we can better understand and support the creation of these marine protected areas, which are also being kind of supported now by these novel finance mechanisms, such as the Debt for Nature swap, which was recently announced by the Ecuadorian government and partners in the last month. So with that, just a last mention to say it's a hugely collaborative work, and it's only possible thanks to people like Salome, who's going to be giving a presentation next, Paulina Separa, who works with us in the taxonomy and the collections, Natalia Tirado, others that some of you probably know. You know, everything that we do is build around and build technical capacity here within the region through these kind of unique opportunities with boats and operations and expeditions like the Atlantis to really bring home science for conservation within the Marine Reserve. And stay posted for what comes next in the summer hydrothermal vents in the Northwest. Okay, thank you very much.
Stuart. Thank you so much. This was fascinating. Like, even after 5 years at CDF, I continue to learn so much. So this was really great. Well, next one is Salome. She's coming up. But I just wanted to quickly say, we did realize that the chat box is not a public feature anymore on Zoom. That's a new thing. So we are producing a QR code for our campaign, and we will share it on the last slide with the Q&A session. And yes, please don't forget to insert your questions for Stuart in the Q&A box, and for Salome as well later after her presentation. So yes, Salome.
Great, thank you.
The floor is yours.
Awesome. I don't know how to turn my video on right now, but it doesn't matter, right? Let me see. Let me see if I can just get my video going. How do I do this? Start video. There we go. Hi, everyone. Happy Oceans Day. It's great to have so many of you connected, and thanks for staying tuned. And can everyone see my screen? Mm-hmm. Awesome. So I'm going to, Stuart just took you to the dark side of things, and I'm going to bring you up a little shallower in our oceans in Galapagos. And I want to tell you about novel kelp forests that we found in the Galapagos around 4 years ago. And this is that is part of my PhD, which is why I'm in Vancouver, Canada right now, presenting you from over here. And all right, let me see if I can get this working. Okay, so I'll just get straight into it. So as Stuart was saying, our deep sea project has been really interested in exploring and characterizing the habitats of deep sea seamounts and and ridges that are around the deepest parts of the Galapagos. But we also do work on the shallower parts, and the square in this map shows you, this is kind of like the shallower part of the platform where our depths are around, vary between 40 and 200 meters. And on this part of the platform, we have what we locally know as bajos. Bajos are like small seamounts or knolls. And when I was part of the project in 2019, this was before any Atlantis was showing up or any of these amazing expedition boats, and I really wanted to get out in the field. So I proposed to use small ROVs to try and explore these bajos that are really important fishing sites. And the Galapagos National Park gave me the approval and they gave me the coordinates, and my goal was to explore these habitats using affordable technology. And this fish that you see here is some of the fish that fishermen typically fish at these bajos. This is el brujo, and if you come to the Galapagos, it's probably the fish that you will see the most on your menu or being sold. So very important commercial fish species here. And the way I did this work was I collaborated with Video Ray, which is a small company that builds small ROVs that can work mainly between like 100 and 500 meters, and got a grant from National Geographic and the support from the Galapagos National Park, and off we went. And so this is what it looks like. So we're mostly from a small screen. We are operating the ROV. We dump it in the water at the site, and this technology there's a lot more luck involved, not luck, but we don't have such precise technologies like on the Atlantis. So often we're just like, okay, this is GPS coordinates, and we're sending the ROV down and let's see what we find. But this ROV still has like a little grabber, so we can grab a few things and lasers, and I've attached some extra cameras onto it. And this is what it looks like on the top side. So we work from the boats that are the regular transport vessels that take you from one island to the other. We call these lanchas de cabotaje. And so in the corner there, you see me with the pilot trying to see what the ROV is seeing. And what you see here is a really long tether, the yellow stuff, which is, we have like 400 meters of tether here, and we manually just like feed tether or rope in which we operate the ROV from. And what we found was we went to some of these seamount locations. Some of them weren't actually seamounts, and that's because we don't have really good bathymetric resolution in shallower parts. And some seamounts weren't actually there where the coordinates said they were, but in other parts we did find some. And we found these cool sponge gardens at about 180 meters and red crustacean coralline algae habitats, which are photosynthetic, which was really cool to see that they can be as deep as 120. And we observed lots of occurrence of commercial fish species. But what I wasn't expecting to find at all was on the last day, which was, this is what the footage when we took the ROV down to the summit of El Bajo de San Luis. And what you're looking at here that's dancing in the currents are kelps. And we were all just flabbergasted because we're not expecting to find large photosynthesizing organisms, which are what seaweed are. And kelps are known for creating marine forests. And as you can see here, we have a marine forest between 50 meters packed with life. And yeah, I was completely shocked and I was thrilled, and so were many other scientists, including Stuart, because kelp is like the mega flora, the charismatic mega flora of the Galapagos, if you're into seaweed. So it's the largest seaweed recorded in all of Ecuador. And like penguins and sea lions and cormorants, they shouldn't be in the tropics, but they are here because we have cold water currents. So it's one of the rare tropical kelp species known in the world. It's endemic, so it's only found here. And we've always known that we've had kelp forests, kelps in the Galapagos. They were found and described back in the 1940s. But something that happened was that in 1982, El Nino event happened. One of the biggest El Nino event happened. And following that event, the marine biologists that have been surveying the Galapagos did not find the kelps anymore in the shallow areas. So it was actually thought to possibly have gone extinct. And this map here on the left shows in brown the areas where Galapagos kelps used to be found. And the green little squares is where the only places where they're found today. And that's thanks to an effort that was done by Stuart together with another group of researchers who were on a mission to finding Galapagos kelps again. And they actually did a really awesome oceanographic model, and where they looked at places where there could be enough light and enough nutrients and cold enough temperatures at depths where Galapagos kelp could be found. And they did find it here on the western part of the archipelago, which is where the coldest water of the Galapagos are. Here, waters are about 10 degrees, can be up to 10 degrees colder than on this side. So when I found the kelp, I was thrilled because I was like, wow, we found a Senia galapagensis right here in a spot where they weren't even thinking of looking. And that's mainly because they didn't have the bathymetry to know that there were these shallow seamounts there. But what I realized was that this kelp species actually looks quite different to the Senia galapagensis. So we realized that possibly what we have here is a new species for the Galapagos and for science, and we had a million questions. So I was like, okay, if I'm going to investigate this further, I might as well do a PhD. And what I'm trying to find out is if this is a new species for science and for the Galapagos, a new tropical kelp species. Are these the last patches, some of the last patches, or is kelp actually really abundant across the Galapagos? We just haven't had the chance to look. And now we have the technology, so it's an exciting time because I think we're going to find a lot more new ecosystems like kelp forests that we didn't know existed before. And I'm also working on a basic characterization of the ecosystems, and I'm studying phenology and seasonality patterns to understand how changes in temperature will affect them, having climate change and future El Niños in mind. And before I went off to do my PhD, I got the chance to collect some really unique, I had some unique opportunities to collect data, a film crew with tech divers. Tech divers are divers who can go beyond 40 meters. They use like 4 tanks, as you can see here, that allows them to go a bit deeper. And they collected a whole bunch of samples for me, which was happy. And as you can see now, this is possibly the largest seaweed for Ecuador. It's definitely the largest ever recorded in the Galapagos. It can be as tall as me. And then I had the really unique chance of going down in a sub with her deepness herself, Sylvia Earle, who is a marine botanist, or what we call a phycologist. And she's actually been amongst the scientists who back in the 1970s collected kelp samples in Galapagos. So when she heard about our discovery here, she was very, very excited and supported our research. And when she brought a submarine for one of her Mission Blue expeditions, she took me down to the kelp forest, and it was really not, obviously it's super cool and everything, but it was a really neat opportunity to see the extent of this kelp forest. It was just football fields of kelp forest on top of the seamount. So not just a little interesting ecosystem, but an important habitat that is probably contributing to and shaping how Galapagos is and why its marine ecosystems is the way it is, especially in terms of productivity. So what have we learned so far? So from the temperature loggers and the data I've collected in the last few years, we know that at least in the central regions, it's colder at these depths by like 4 to 5 degrees, which is possibly one of the reasons why we find it at this depth and we haven't seen it shallower. There's very, very little light down here, but there's still somehow photosynthesizing, so that there's less than 1% of radiance light. So if 1% of all the light that hits the surface of the ocean, less than 1% is reaching at this depth. So somehow they're doing magic photosynthesis. But the kelp forests have a good density. They have a high density, which qualifies them as forming a kelp forest. They're reproducing. This dark patch on the leaf is what we call a sori, and that is evidence that they have reproducing. And this is a juvenile, so they're in a good, healthy state. And if you're more interested, we've published a paper on presenting all the physiological features of this kelp species, and we need a bit more genetic work to be done to be able to determine if it's a brand new species or a very different variant. So stay tuned. And just for comparison, that is Eisenia galapagensis, and this is the new Eisenia sp, so you can see the difference. And the work that we have going on in progress right now, or I just actually came back from being in the field, is I'm studying the abundance and distribution. And so we're going around, I've got a new ROV, and I've been going around the island opportunistically trying to look for more kelp forests, and we have found more. This is one of the Isla of Santa Fe, and it's made up of this new species that is huge. In fact, the ones in Santa Fe are larger than the ones in Bajo San Luis. And we refound the ones that Stuart and his crew found here in the western part of the archipelago. So I've included them in my study so I can do some comparisons between these different types of kelp forests. And just again to show you the comparison of these kelps. And so what we're doing, so what I've been also doing is I've been going back with the ROV, and I've been surveying during the warm and the cold season just to understand phenological processes. And I use stereo video technology. That's why I have the two cameras here that film at the same time. And using special software, I'm actually able to measure the exact size of the kelps. And this allows me to do a population structure. And so stay tuned. I'm hoping to be putting out a lot of data, new data soon about the ecology and composition of this kelp forest. Other ways we collect data about understanding this ecosystem better is we send down a CTD. So Stuart showed you this like epic CTD Rosetta that the Atlantis has. But my type of work keeps you really fit. So it's kind of like doing CrossFit. You send the CTD down, and then we all have to haul it up, and we feel very strong and muscular after, because it's pretty heavy gear. And that's how we collect data. So that is, we actually are also able to collect samples with the ROV, with the grabber. This was a few months ago when we were successfully grabbing kelps with the grabber of the ROV. Stuart and I collecting water samples as well to study the nutrients. And here's a short video of how you learn, how do you grab. So you first practice in a pool. And this was my first ever attempt of trying to do it myself. And it was really hard. And mostly I'm at the top of the boat trying not to vomit because it gets really seasick. And but it's not impossible. I managed to grab it, and then I had to like yank it up, and then you see the holdfast, which is the root system detached. And then a bloody sea lion wanted to come and steal my sample, which was all fun and beautiful, but honestly, I was going to be very angry if it had managed to take the only two samples. But yeah, and this is the type of smile you get when you succeed on this. And what I do with these samples is we get a lot of data from them, mainly morphometrics. But a cool thing that we are trying to do is we actually study them like trees. We cut the stipe and we count the growth rings in the stipe to determine how old they are. And this gives us really interesting information because I want to know how old the oldest group is. And that kind of gives us an idea of when was the last time they all died off. So for example, maybe if everyone is as old as the last El Nino, we know that during El Nino, there might be major die-offs. And that's something that I'll be putting out soon. And finally, just to highlight why this work is important, what we've learned is that the mesophoric zone of the Galapagos hosts really critical foundation species. Kelp is a foundation species. It provides food and habitat for many other species. And it's really interesting to now know that there are many more located across the Galapagos. And this is the current marine protected area scheme. And the red zones are where our current no-take zones are, where no fishing is allowed. And the green is the future ones that are proposed. And if you notice, two of the kelp forests are not inside of this, these proposed protected areas. And this is why this is really valuable information for the Galapagos National Park, because now that they know to consider these sites for future zoning schemes or other types of marine protected management schemes that will allow these ecosystems to become protected. And up until now, they were invisible to us. So when we don't know that something exists, then how can you protect it? So this is why this work is really important. And also during this exploration, it closes key knowledge gaps about the different distribution of biodiversity and the connectivity between shallow and mesophoric ecosystems. We found that many of the fish species in our ecosystem, in the kelp, are the same as the shallow coral reefs that most tourists go and swim out. So there's definitely connectivity there between the kelp forests and the habitats that we know more about. And yeah, the technology is there now. It's an exciting time to be a marine biologist. If there are any listeners there who might be interested in going into this field, the technology is cheaper and it's getting better, and you can actually be a true explorer if you want to get into marine biology, because there's a lot to discover. And yeah, that's it from me. And this is a photo this is a photo of Paulina, who's one of our research assistants, and she's proudly holding a kelp forest on an expedition that she had to lead because I got sick that day, and she made us all very proud. And I want to give thanks to the many, 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 many people who thank you. Tuned. Thanks for listening, and we're happy to take questions.
Perfect, Salome. Thank you. No worries about going over time, because I think it was very interesting. So we still have all the participants here with us.
Yeah. Do you want me to stop sharing?
Yes. So Camila can share her screen again. Wait for questions. There is nothing in yet.
What?
Yes, everyone ask questions, but meanwhile, maybe I can ask a question to fill the time. So yeah, because it is known that the 2023 is going to be a strong El Nino that is coming, right? So how are you planning to do the monitoring, and how long might this period be? And how are you going to be?
Hello?
And so I didn't know if El Nino was coming, but I think if with the opportunity, the least we should do is go back during ideally the same times that I went before, so during the cold season and the warm season, and monitor the conditions. And thankfully, we're hoping to get another small ROV. The ROV that I work with belongs to the university. But this is the great thing of small ROVs, that if we can just send them down as a pair of eyes and just check that the kelp forest is still there, that is worth doing, because this kelp forest could probably like die off in the wrong conditions, probably like within a month's time. So if conditions are bad, it is possible that we go down there and it's a bare place without the kelps. And it'll be really interesting to see. However, unlike corals, kelps are actually not the canary of the mines. They are pretty resilient. They grow much faster, and they have a seed bank. So I want to give a positive message out there, which is that these are resilient ecosystems, and I think that they are going to be around. And if El Nino comes, they might disappear, but I think they'll make a reappearance after.
That's good news.
Thank you. I'm right, but I need to go and collect the data to do that.
So yeah, we have some questions here. Peter Kramer is asking if you can please repeat the info, how these two species of kelp are distributed within the islands. Thank you.
Sorry, I missed the first part. Oh, is the question there in the oh.
Yes, if you can just repeat the information that you already mentioned.
Oh, sure. Should I share the screen again?
Oh yeah, maybe.
Yeah, I can do that. So how these are, okay. Do you see my screen? Okay, oh, that's not the screen I want. So that's a good question. So in this map here, you can see this is where the Eisenia galapagensis is, which is the...
Oh, sorry, we're not seeing your screen. I think Camila, you have to put your screen down maybe.
But yeah, in short form, the Eisenia galapagensis, what I call the OG kelp, is located in the western part of the archipelago, so off the coast of Isabela and Fernandina. That's where we found it and the efforts we've made. And The novel kelp species are found in the south central region. So
Oh, here we are, yes.
yeah,
We can see it now.
so this is where we found the novel kelp forests. So they're usually an offshore, either on the top of this seamount here, or on an island continental slope here, between 60 meters, here between 50 meters. Whereas the OG kelp, Eisenia galapagensis, is mostly found in shallower water, actually between 20 and 30 meters, here off the coast of Fernandina and Isabela. Do you have more
Perfect, thank you. We just had a comment from Sylvia Harcourt. She says, no questions, but just to say thanks so much. Fascinating, congratulations. And she unfortunately had to leave the presentations. So should we wait a little bit longer? There is no...
I just wanted
here
to
we
add
go.
or tie things back into like the study that I'm doing, the study I presented and what Stuart presented. I think as an ecologist, you think a lot about the processes of what's happening here, what we're finding. And when I see the, I'll stop sharing here. When I see the coral reefs that Stuart found and with Michelle during the Atlantis, those huge reefs, they're all filter feeders, right? They're just filter feeding. And obviously then the Galapagos, they're able, one of the reasons why I think this reef is so big here is there's all this food, this what we call marine snow that they can filter feed. And I'm in my head, I'm imagining that the kelp is producing lots of food for them to eat as well. So I think we'll need to find more, we'll need to collect more data, but I think there's a connection between the kelp and its leaves becoming eroded and feeding this giant reef. Because one of the reasons we haven't found this kelp before is they don't have floaters. Many kelps, and for example, California float and beach. So you find kelp at the beach. We've never seen this kelp before because it doesn't, it just sinks into the deep. So my theory is that as these kelps die off and sink into the deep, they're being devoured by all of these filter feeding invertebrates. What do you think, Stuart?
Yeah, I mean, the islands, as we know from, I mean, there's a huge amount of work which we've done in the shallow areas and rocky reef around the islands. And one thing which is clear is that it's an amazingly dynamic and productive system. So the turnover of everything eating everything is incredible in the islands. And it's one of the reasons why it actually has this kind of resilience. You know, it's been hit by these very strong kind of El Nino signals for thousands of years. And a lot of the system, a lot of the communities, the marine communities are configured to basically sort of survive within that. So, you know, their ecological niches kind of falls within this kind of dynamic, this dynamic system. But what we're looking at the moment is something slightly different, because as you move down into deeper water, there are different types of disturbances. And there's also kind of potential that certain areas are less disturbed compared to the surface during these very kind of strong climatic events historically. I saw within the chat there was a question on any ideas why the deep coral reef that we found, which was one of several during the cruise, might be a particularly kind of important coral hotspot. And it's, I mean, in part, the oceanographic features around the islands, the upwelling into the surface, that provides these kind of huge productive patches of water, which are associated with that kind of physical mountain. I mean, all the water is being pushed up over the top. And because of the way that these things work, the oceanographic, the current, the oceanography of these things, the current kind of local circular system sometimes, where you tend to get whatever's produced up there kind of stays up there, and as it falls, as Salome mentioned, you know, it's providing kind of like this big kind of nutrient fall into an area where seafloor rises enough to be able to support ideally kind of these coral frameworks. A lot of this is to do with how coral, kind of the calcium carbonate and the aragonite, that's the skeleton of corals, can actually form in the tropics, because usually it's limiting. In places like Galapagos, it's quite limiting. And that's kind of interesting for us as well, because it shows that, you know, there's, with acidification under ocean warming scenarios, you also tend to get these dead zones within the water column as well, which also kind of hugely affect distribution of these, where these reefs can form. So this kind of shows that if you don't kind of drive destructive bottom trawling fishing practices across a reef and leave it for several thousands of years to do what it does best, you can have these kind of hotspots, which are important source areas for these amazingly productive species, which basically foster and harbor this amazing 3D complexity within them for thousands of other species. And most of these, you know, we're only starting to register now, and it's really kind of bringing a whole new dimension. So we can start to model that and then look at similar conditions across other parts of the archipelago. But, you know, to do that, we need to start getting the base bathymetry to be able to detect them, which is what we found during this cruise. And hopefully we'll continue to do that now.
So we have a new question. It says, is there much temporal and seasonal variation in the deep currents?
They are believed to be reasonably consistent in other places. And, you know, one of the problems that we have here is that to be able to get that deep to do these surveys, I mean, Salome showed, you know, working up, having a good workout with that CTD instrument on a metal frame, that you can lower those by hand. We've done that kind of jury-rigged oceanography in the last 20 years with the park, and you know, we had 3 people pulling these things up and down 100 meters. Imagine these things have to go down to about between 2,000 and 3,000 meters. So again, you know, we have very limited snapshots of what's going on down the bottom there. So we need more information basically, but a lot of the observations are that the currents are lower as you get down, but they switch as you go down as well, because the subcurrents interact with the surface currents. So short answer is that we suspect that the currents are affected by seasonality, because that affects in the surface production, and what happens down lower also affects how the undercurrents interact as you go down. But we don't know all the details yet.
Perfect. Thank you. Well, we got a lot of messages that it was a very inspiring conversation. So thank you so much. I agree. And we're already getting in some donations as well. So thank you for everyone who joined and who is supporting us. And we will be sharing the recording with you as well as the link. So hopefully you can support the project by next Friday, and we can raise funds for this workstation that we need to analyze all this information that we currently have. Thank you, everyone.
Thank you, guys. I think we've reached the end of the webinar right now. We still invite you to keep on looking for the bi-monthly newsletters and all our updates on social media, because we will be sharing new information as soon as we get any discoveries and the information from our scientists. So thank you, guys.
Great. Thank you very much, everybody. And if you have any particular questions, we're very happy to follow up.
Yeah, we have one last question before we leave. Is it possible to install sea bottom cameras to learn more about kelp habitat? That's probably for Salome.
Sorry, let me read that again. Is it possible to install? Yes, it's very possible to probably drop some. What becomes impossible is to find them again. And this is one of the conundrums of working in the deep, right? Like it's much easier to locate. It's already not easy finding things to scuba diving because we just don't have GPS underwater. So you can do this, and usually you would just need more than to have like some kind of system that either releases itself and comes back up to the surface so we can collect it, which is what drop cameras are. There are drop cameras out there that you can drop and then leave them there a few hours and then they come back up. or you have a system in which your ROV can, like through radar or some kind of detection, easily find the camera again underwater and then grab it. But it's the type of experiment that needs a lot of funding to be done, because you're highly likely to lose a lot of expensive equipment at the initial phases. But this is something I have been thinking about, because I want to creep on the organisms that use the kelp forest, and the best way of doing it is kind of hide cameras and see who's doing what. And so far, the ROV does tend to like scare off some fish, so we have a bias of recording species that are not too intimidated by a loud moving ROV. Great question.
Okay. Well, thank you so much, guys. See you next time.
Have a good day, everyone.
Thank you, everyone.
Great. Thanks, everyone.
Bye.